glycerin or CGP administration to induce a pain phenotype. That's mainly done in Copenhagen. And the reason why they think that FHM is not a good model is because if you give patients CGP, it doesn't work.
Yes. But that's probably...
I want to know from you whether you think that why this is. And does that say that FHM is not a good model for migraine? Or is it because it does exactly what your model is that it does?
Yeah, it seems that if you apply more CGRP, it does nothing because pituitary receptors are already upregulated. But in other types of migraine, it would just reach the stage that is already reached in FHM1 patients.
Yeah, there's still a problem, and then I go to the next phase, is P2X3 is continuously upregulated.
Not in Europe.
Yeah, but if you think that that's relevant to migraine, right, even for patients that do not have a calcium channel mutation but still have upregulated CGRP, would you think that, what is then the, how would you then define the FHM1 mouse model? Because it has a continuous upregulated P2X3 receptor. It is a chronic migraine model, would you?
Yes.
How would that show in mouse?
In terms of behavior or...
Exactly. P2X3 is always upregulated. You cannot even regulate it in CGRP, with CGRP, right? It doesn't, is not modifiable with BNP anymore. Yes. So it's always upregulated. If this nothing model is relevant to the patient, what would the patient then have with respect to the phenotype? Is it bad to have always upregulated P2X3 receptors?
Yeah, it is, because it also elevates the excitability of neurons that in turn can lead to higher activity in trigeminal neurons, so higher probability of painful stimuli to go through trigeminal neurons and come to higher brain levels, and that
so you would not predict perhaps that these mice and patients with these mutations are in constant pain, but you would think that any small disturbance will be really painful.
Yes.
Is that correct?
Yes.
Unfortunately, that's not the perception in patients. So that's how I struggle, how to reconcile it.
But it could not mean that any non-painful stimuli becomes painful, but it could promote a seizure. So probably even like if cortical-suppressive depression occurs, it induces the activity of trigeminal ganglion neurons, and if they are in the downregulated state, that could just lead to nothing. But if they're in upregulated state, that could lead to real seizure and to pain perception in cortex.
Yeah, so the question that we have to do in the animals, in the patient studies, is to find out what the stimuli are.
Yes.
Right? What are normal stimuli? We did so many things with other, let's say, researchers to find a stimulus for a painful attack or a migraine attack or a circulating attack, and we couldn't find it. So, to me, at the moment, the trigeminal ganglia phenotype is a little bit isolated from understanding the bigger picture.
Yes, but
it's just a challenge. It's not criticism. It's a challenge.
Yes, but it's always like that because you need to first study something and then you implicate everything into the global picture.
Good. I would like to go now to the exo-experiments, and you can put one of these slides on there where you have the Western blot sensor. Maybe one back or few back.
So not excitability?
No. One with a lot of such something like this. So this is a little bit unclear from reading the thesis. So you do experiments in individual neurons, right? Often 30 to 40, 50, 60.
Yes.
And you do Western blotting,
Yes.
right? And I have two questions, or a few questions. One is, the ends for the Western blot are very small, 2 to 4.
This is the experiment.
Yeah, I understand. But you draw for a conclusion from those. So I have two questions. What about the statistics and how you deal with that? And second is, which picture do you take for these nice graphs? So what's the variation in these pictures?
Yes, yes.
And I'm worried about that throughout all of the different experiments.
So for Western blot, basically, this is a mean picture with the variance. This is standard deviation, and this is 3 or 4 experiments. And this picture is a representative.
Yeah, sorry. I understand everything, but you do this with ECL, right? So the exposure is very critical. So if you have a higher background, lower background, so how do you actually do the experiment? Because the standardizers are very small.
Yes.
I'm surprised, given a little bit of the background and the difference in the background. And also how the, look at, for instance, the Western blot, where you have, oops.
Here?
No, the one really wasn't prepared down.
Probably this is the first one.
Well, that perhaps.
Right?
It doesn't matter, because on the right where you have the lithographs and non-lithographs, you have quite a lot of backgrounds, right? Which is still surprising how you can get such a small standard deviation in the graph itself.
Background, you mean?
Backgrounds and the smearing of the bands.
This?
Yes, a little bit of smearing on the right there is already giving a signal. So I was wondering how you do these experiments.
Actually, as I mentioned before, I personally didn't do this experiment. These experiments were done by Sandra Velotti. And I'm actually not sure how to answer this question because I did not analyze this data per se.
Okay. That's a fair comment. And the comment for statistics is whether they are independent variables, so biological replicates, or whether they are technical replicates, where you reload the sample, let's say several times, and average this.
Yes. As I understand, you load the gel several times and then calculate the mean and deviation.
I have to go to the next point because what you say now is not what you wrote down in your description, that you reload it, and then so you have a technical replicate and a biological replicate.
No, as
you didn't do that. Okay. So the reason why I'm asking is because this is such a central theme in the experiments, in the different papers, that I would, out of curiosity, want to know how you deal with it, because it's a sometimes very minor effect that feels statistically significant, and draw a quite strong conclusion. Right? So that's why I wanted to know. Also, because you make lysates, the second question. Because you make lysates, you lose a little bit of the biological context, right? In the sense that you don't know how many cells, let's say, have, for instance, the upregulation of the, in this case, what I call it with the serum phosphor antibody.
Yes.
Any idea whether how this relates? So if you have few cells that have a lot of phosphorylation and all of them don't, or have all of them a little bit increased or decreased phosphorylation level.
But usually the level, the amount of tissue is similar because you also normalize it on the total number, and you see no difference in the tissue.
Yeah, but then you still lose the cellular contact because you lump all the cells, you make a homogenate, and you don't know whether there were certain cells that express serum phosphonates, others that don't. With the BNP immunohistochemistry you showed, there were only 2% that showed an effect. Then I want to know whether this is increased in level in the mutant or in the number of cells that are increased. So that's the idea.
Yeah, this is the question because we didn't do it in the phosphorylation.
Now the real question is, can you come up with an experiment how you studied it in a different way? How you report an assay that shows fluorescence, for instance, in the cell. So you can look at the cell basis. Is there a fluorescent assay, report assay that you could use to prove what you showed is false?
I don't think there is I could be mistaken, but I don't think there is such a say for phosphorylated.
Or any other thing. So, for instance, that you can show that the receptors indeed go to the non-lipid rafts and are phosphorylated at the non-lipid rafts or dephosphorylated non-lipid.
Actually, they are. We showed that these two processes are separate, and receptor phosphorylation does not mean that receptors are in lipid rafts or that they do go to lipid rafts. Phosphorylation occurs in both raft and non-raft compartments equally.
Yeah, but we have to prove that. You don't know that if you make a light it, so you right? Is that correct?
Yes, but then we showed that if you completely destroy lipid rafts, an enzyme is still able to upregulate serum phosphorylation. But since there are no cells in lipid rafts, then you can only upregulate downregulate serum phosphorylation in non-raft compartments, meaning that it also affects non-raft compartments. And this upregulation is exactly 2 times less than in the whole situation, meaning that pretty much it affects equally these two compartments. I also have a slide for it.
No, this is a fair comment. The last thing is you study very young trigeminal ganglion, right? In 12 and 14.
Yes.
In one of the papers, P30 is the latest, I think. You call P30 then adult.
Yes.
Everything begs for the question, how is that related to myopathophysiology, where this is a process that starts later in life, and P30 is maybe not the latest time point that you should study. Is there any evidence that there is a regulation over age of CGRP mechanism or the BNP and NPRA mechanism, so that you get a totally different result if you study mice of 3 months, 5 months, 6 months, 8 months?
Yeah, we only showed in real adult mice, the expression of either BNP or NPRA.
But that's P30.
Yeah.
That's very early, because that's the P30 are mice that I don't even see the experiments, because that's the time that we start preparing them for experiments. Our experiments are P60, P90, P120, so wait much later. I asked the question because another researcher find less evidence for a difference in CGRP content of trigeminal ganglia, but they study older mice. So the question is this all relevant for the moment for quite young animals? And will this perhaps different when you study older?
Yeah, we don't know yet because we haven't done any experiments in older mice. So this is probably.
Do you know if you look at the disease, the development of the disease? So, any idea what happens in let's say, when patients go to the 40, 50, 60? What happens with the development of the disease? In females and in males.
You mean if the disease originates later?
Or what happens and whether it's relevant to look also at all the later time points because the disease changes.
I'm example, when the menopause already occurs, you can expect less frequent attacks or even complete stop.
That's exactly my point, because later in life the attacks stop, right? After menopause, the attacking females tend to disappear. The question is whether the trigeminal ganglion system has adapted that and is, let's say, helping in a way.
This is a very interesting.
Right. So I was wondering whether your results and your experiments could tell it. If you only have to study that later in life.
Yeah, you need to study all the months.
Minor thing, but I could not find a gender in any of the studies. So is this male mice or female?
As I understand, this is a mix. So,
Do you think that's a good idea?
Since
Look at the graph, because if this is a mix, roughly you know whether it's a good idea or not to mix them.
in which part?
And of the arrow bars. Look at the arrow bars.
Considering that the disease occurs more in females, probably you could study just female ones. But to get an idea of the whole population, probably that's not such a bad idea to study both.
No, yeah, what I was referring to is that when the error bars are so small, when you mix males and females, apparently there's not something different between males and females, otherwise you'd have a bigger error bar. So apparently in this case there's not so much of a difference. The question is if you study all mice, whether you look at the difference, and you should have studied that or be more careful.
Yeah, that's another point.
Good. I move to the, let's say, more specific part of the PTH3 receptor and the BMP and NPR-A system. PTH3 receptors are ATP gated, right? So you think ATP is important. Where does the ATP come from in migraine? Why is this an important system to begin with?
It could be released from synapses with other mediators, like peptide mediators, and it also could be released from non-neuronal cells, especially during inflammation. And since cortical spreading depression was supposed to affect this and could increase inflammatory response, probably that is the region from where ATP comes from.
Yeah, what you in one sentence you make very clear is actually a very, let's say, is a hypothesis that not too many in the field are, let's say, agreeing on. So that you think that, and I agree, so so you think that inflammation in the brain is important in migraine.
Yes.
That was studied, neurogenic inflammation was studied in the, let's say, a long time ago. It was totally banned. Nobody was allowed to mention the word inflammation for, let's say, a decade or so. And now with the Courageous paper that you also had, with the connection activation, you see some inflammation coming up. But in the meantime, your data were one of the few that and the one of Cerruti and Marchenkova one of the few to show that inflammation indeed may be important in migraine. So you think that although we normally have ATP in really inflamed, injured tissue, this is also occurring in migraine, but at the other level. Is that correct? At the less level, there's obviously no injury.
Yes, yes.
It's not a nerve lesion or something.
No injury, yeah, but still I think upon activation after cortical spreading depression, this inflammatory signal could promote ATP release.
Do you now understand why I asked you the question, what is going before the general ganglion? So in this case, in cortical spreading depression, you could have an inflammatory reaction. That inflammatory milieu affects the trigeminal ganglion. So that's why you think the noxious stimuli come from. Is that correct?
Yes.
Okay. What about the role of PTH3s in non-trigeminal ganglion cells? On page, what is it, 61, page 69, you discuss DRGs and brainstem trigonal nuclei. Right? What about T cells in relation to what you showed?
In relation to migraine.
And in relation to migraine. Does that change your picture about migraine if you would have studied DRGs in the same way you treat tangent ganglia, or if you would study brainstem nuclei?
So in dorsal root ganglion, for example, there are of course also P2X3 receptors, but it was shown that there is less P2X3 receptors in DRG and more P2X3 receptors, so heterometic receptor types. So it's not very clear if the same mechanisms could be applicable to DRG. Perhaps they could, but since there are less receptors, probably they don't contribute that much to the development